My name is Bruce Stillman. I'm president of Cold Spring Harbor Laboratory, and I'm also a professor at the laboratory, and today we're going to talk about research in my laboratory. So part of the work that's going on now in the laboratory focuses on yeast replication and identifying the proteins that are involved in the process of initiation of DNA replication, the detailed biochemistry of those to really understand how these proteins work. And we're carrying this to the structural level in collaboration with Hu-Ling Lee at Brookhaven National Laboratory, who's a cryo-electron microscopist, in collaboration with Lamore Joshua Torr's laboratory, and in collaboration with Christian Speck's laboratory. Christian now has a laboratory in London. And we are interested in identifying the kind of atomic details of how this process of replication actually occurs. The other aspect of it that's going on in the laboratory is to focus on the human chromosome replication, and particularly the very diverse roles of ORC. One of the things that we've done is just most recently has shown that the origin recognition complex, as I said before, controls not only DNA replication but centriole duplication. The centrioles are the organelles that organize microtubules for chromosome segregation in mitosis. In the absence of the ORC proteins, particularly ORC1, the process of centriole duplication goes awry and you get too many centrioles, and this causes mischromosome segregation during mitosis and genetic defects. In fact, mutations in Meyer-Gorlin syndrome affect that process. And we published that a number of years ago. Now we have also shown that ORC does this, the origin recognition complex does this by interacting with an important and very key regulator of the cell division cycle called Cyclin E-CDK2. Cyclin E is one of the many cyclins in cells, but it is the one protein that is actually dysregulated in almost every cancer. And these Cyclin E proteins, they're very important because not only do they are involved in formation of the pre-replicative complexes that establish chromosome replication, but they also control the ability of centrioles to duplicate once and only once per cell cycle. And when Cyclin E levels are messed up, cells make errors in both of these processes, in the process of DNA replication causing genetic damage, and also the process of mitosis through the centriole defect, which causes things like aneuploidy or chromosome imbalances. These are very characteristic of cancer cells. So most recently we have shown that ORC1, the large subunit of the origin recognition complex, directly regulates the Cyclin E-CDK2 kinase activity, but also directly regulates the transcription of the Cyclin E gene. And it does so by interacting with the famous retinoblastoma protein, which is one of the first tumor suppressor proteins that was identified, and also a protein called a histone methyltransferase that causes methyl groups on the histones around the promoter of the Cyclin E gene, and that forms a highly repressive chromatin state and localization of that DNA in the nucleus that the genes are not expressed. The Cyclin E gene is not expressed. This is important because it's uncovered a process that we have recognized now in the decision of whether newly born cells, a cell that has just gone through mitosis to produce two identical daughter cells, whether those cells to go on to proliferate or not. The cells have a choice. They can go on and produce from 2 cells to produce 4 and 8 and 16, etc., or they can choose based on biochemical information to not proliferate and go into a period of what we call replicative senescence or quiescence. The quiescent state, this decision of whether to proliferate or not, is very, very important during development in organisms, and certainly in multicellular organisms, and is controlled by extracellular and intracellular processes that measure the energy and the nutrient and also the growth regulatory processes that cells have to respond to. So when a cell goes through mitosis, it has to shut down temporarily the decision-making process of whether to proliferate or not, and that really involves the decision of whether to control cyclin E expression. So the process of making a decision of whether to divide or not to divide is a very important process in cells, and we've shown that the large subunit of the origin recognition complex is involved in this, along with the retinoblastoma tumor suppressor protein and the histone methyltransferase SUVA H31. These proteins form a repressive complex on the cyclin E promoter, and interestingly, we also have published that ORC1 associates with mitotic chromosomes as cells go through mitosis. And then those chromosomes are inherited and decorated with ORC1 associated with these mitotic chromosomes, and ORC1 can then participate in the process of forming a repressive complex on the cyclin E promoter gene, the promoter of the cyclin E gene. And this repressive complex provides a period in early G1 phase when the cyclin E protein is not expressed. Furthermore, ORC1, if there is any cyclin E in the cell, ORC1 actually inhibits the kinase activity associated with that cyclin. And this provides a window in early G1 where the cyclin CDK2 is not active. This enables the cell to make a decision of whether to proliferate or not. And this decision is really at the core of development, that cells, newly born cells, have to go through a process of deciding whether to proliferate or not, to go into multiple rounds of proliferation and to amplify the tissue volume, or to go into a replicative quiescence and not proliferate, and this establishes tissue homeostasis. We believe that ORC1 is involved in that intimate decision, and also it provides this window where the cells can integrate all of the extracellular growth signals and nutrients to then make a decision of whether to proliferate or not. That is then triggered by many signaling cascades that focus in on this series of transcription factors that are known to interact with the retinoblastoma protein called the E2F transcription factors. And what ORC1 does is to control the expression of the genes that activate the E2F transcription factor pathway. So this is an interesting finding because it shows that a key DNA replication protein is involved in that decision-making process. But what we've also shown is that the protein called CDC6, which is another initiation of replication protein, which is highly sequence related to ORC1 and interacts with the origin recognition complex, that protein actually antagonizes the repression and in fact activates the cyclin E gene. So you have a situation where there are opposing roles of these two initiation of DNA replication proteins, one repressing cyclin E and one activating cyclin E. And it turns out that they are expressed at different times during G1 phase of the cell cycle. And they then are involved in this decision of whether to proliferate or not. And so this is an interesting link to the process of actually going through DNA replication to the process of deciding when or if you're going to do it. And we're following up on this because we think this is just the tip of the iceberg of the role of the origin recognition complex in understanding actually lots of things that are going on in the nucleus, not just this decision of whether to proliferate or not. I suspect that ORC is involved in whole nuclear structural, the structure of the entire nucleus and how the DNA is folded in the nucleus, and also how other processes such as other aspects of gene expression and other aspects of cell proliferation, even the establishment of pre-replicative complexes themselves, is controlled by this organization of the nucleus.